Good morning, students. Today we will start our genetics class, and we will start with the topic genetics, introduction, and history of discoveries in this field. So, this field of genetics is an age-old field of study where different scientists have performed several work and they have come to a conclusion of mind-blowing discoveries. So before going to the deep study of this particular field, we need to know that what is genetics and its historical perspective. So let's begin our today's class.
So now we just go for a brief definition of what is genetics. So the scientific study of it is a scientific study of heredity. Genetics pertains to human and all other organisms. So for example, there is a human genetics, mouse genetics, fly genetics. So heredity. What is the meaning of heredity? The meaning of heredity is transfer of genetic traits from parents to their offspring. Whatever the case, if the parents is human, then also the genetics applied to the humans, and in case of the other organism as well as bacteria also. Genetics refer to the transfer of genetic trait from one generation to the another. Fine. This is the reason why we find similarity of a child with their parents and from their parents to their grandparents. So this is a way or flow of genetic information which carried out generation after generation. Now, if we look at the classification of genetics, we can divide genetics into 4 different classes. The first one is the classical genetics. So classical genetics is the branch of genetics based solely on visible results of reproductive act. It is the oldest discipline in the field of genetics, and this field of genetics is established by the father of genetics, Gregor Johann Mendel. We will go some brief story about the Mendel in the later phase of this class. Next is the molecular genetics. Molecular genetics is the study of molecular structure of DNA, its cellular activities, and its influence determining the overall makeup of an organism. So as we all know, the basic genetic material of every cell is DNA. So that is why the study of DNA and its characteristics is called the molecular genetics. Next is the population genetics. It's a subfield of genetics that deals with genetic differences within and between the population and is part of evolutionary biology. Studies in the branch of biology examine such phenomena as adaptation, speciation, population structure. Like a particular species, if we all know the term Pangea, that is the ancient form of earth. So in that case, no Himalayas were there, and the species are able to freely move from one continent to another. After that, when Himalaya mountain evolved or formed, then these two species get separated by a barrier. That barrier is a natural barrier, and that barrier is the main cause of the difference of the climate between two places, which was initially the same, and the species are also same. And after the development of Himalayas, after the development of a barrier, the two species are getting differentiated and differentially evolved, and this is the example of the branch which deals with the population genetics. This can also apply to the human also. Now, if the final or the recent form of genetics is the quantitative genetics. The quantitative genetics allows one to estimate heritability, that is the ratio of genetic variance and total phenotypical variance, or the ratio between genetic variance arranged solely from additive genetic effects and total phenotypical variance. So, before going in depth with this particular subject, we need to know or we need to understand some basic questions. So, the first question is, why is genetics important to my family or at all me? So, genetics is able to explain some of the very important questions of ourselves. That is, what is the basic phenomena which makes us unique? I am the only person who is a unique of my way. Is it only the developmental aspects, or some genetics aspects are also there that we are able to know if we go through the genetics of me and my family? Why family members look alike? A particular child have a similar characteristics with his father or his mother. It may be possible that the eye of a particular child is resembled with his father, but the lip is resembled with his mother. So this deals with the genetics. Why some disease like diabetes or cancer run in the families? Now we all know that cancer and diabetes, both of these severe diseases, have the role of genetic predisposition. So genetics have the role for the development of this kind of diseases. So if we are able to know that, we can restrict ourselves or we can take some measures to prevent these kind of diseases. How learning your family health history can help you stay healthy? This is the thing. If we know my family health history, that if somebody of my family have any kind of diabetic history, then I may take precautions. Fine. Now, what makes me unique? What is the basic thing, a basic biological molecule which makes me unique? And as you all know, that biological molecule is gene. Gene is the main source of information which give instruction to the rest of the part of the mechanism, body mechanism, and control it. Fine. And through this genetic control, it is determined that how we look and how our body works and etc. Since each and everyone have a slightly different genes, everyone also have a different set of instructions. Like in each and every human being are 99.9% similar, but that 0.1 or 0.01% dissimilarities makes us unique, and that dissimilarities are basically present in the genes. So that is why genes are the basic reasons or the one reason which makes us and you unique. If we look some little bit deeper into the characteristics of genes, we may found that each and every individual have 2 copy of genes in its cell, and a particular individual have billions of cells. If like us humans, we have billions of cells in our body, and each and every cell contains same set of DNA. and same set of DNA contain the same set of gene. So genes are arranged in structure called chromosome. So chromosome is nothing but a modified structure of gene, and gene is present in DNA. So in that case, chromosome is basically made up of DNA, and DNA is composed of multiple genes. And human, each and every species, each and every organism have different set of DNA and different set of or different characteristics of gene, but in human, and their gene number is conserved among the species. Like in human, we have 23 pair of chromosome, and among that 23 pair of chromosome, 22 are autosomes and one pair is the sex chromosome. Fine. So that 23 pair of chromosome is actually present in each and every cell of our body. So that is why if that particular chromosome or DNA comes from our parents, my father, my mother, so that means I might have some similarity with them. That similarity may be a combination, or that similarity may be little bit dominant towards one parent. That is a different set of question. But we must have some similarity with our parents, because we share the similar genes. It's not the same. We don't share the same gene. It's similar because, as I already mentioned, the 99.99% genes are same in each and every human, but that 0.01% differences which have, that makes us unique. So that means we have similar kind of gene. Fine. So now genes can also increase the risk of family for getting certain health conditions, because as a family we share some habits like diets. We live in a similar kind of environment. So this diet environment, that can also influence our genetics. We will study these things later in our course. So if it is possible to know my family history or my family health history, does that help me to stay healthy? Yes. Identify the risks due to shared genes like hemophilia. Now doctors suggest that before marriage a couple should go for the test of hemophilia, whether they are hemophilic or they are carrier for the hemophilia genes. So by this way, this can be easily determined in another way also. If I have some hemophilic history in my family, so there might have some probability that I am a minor hemophilic or a carrier for the hemophilia. So that is why knowing family history actually helps us to identify the genes, to change our lifestyle, and in this way we can prevent the possibility of the occurrence of some disease, and we can remain healthy for a relatively longer period of time. So now we are just looking at a glance. There are different branches of genetics. The number one is the classical or formal genetics, which is the we will start our course with the classical genetics part only, and that part actually mainly restrict with the Mendelian genetics. Clinical genetics, the diagnosis, prognosis, In some cases, the treatment of genetic disease. Genetic counseling. An important area within clinical genetics involves the diagnosis, risk assessment, and interpersonal communication. We can assess the risk, the occurrence of some disease by counseling them. Cancer genetics, which is a very emerging field of study. The study of genetic factors in inherited and sporadic cancers. Cytogenetics, the study of chromosome in health and diseases. As we know, if some damage in chromosome occurs, that may lead to the development of several diseases. And cytogenetics is a field of study which deals with this kind of study of chromosomes. Biochemical genetics, the biochemistry of nucleic acid and protein, including enzymes. We all have the idea of enzymes, and the study of the regulation of enzymes for its activity is basically comes under the area of biochemical genetics. Pharmacogenetics, how genes govern the absorption, metabolism, and disposal of drug and towards reaction to them. Molecular genetics, the molecular study of genetics, including particularly DNA and RNA. This includes replication also, transcription also. Immunogenetics, the genetics of immune system, including blood group, HLA, and the immunoglobulins. Behavioral genetics, the study of genetic factor in behavior in health and disease, including mental retardation and mental illness. Some kind of mental retardation and mental illness, including depression also, have to have some or might have some genetic history. So this comes under the area of behavioral genetics. Population genetics, which is a very interesting part of study, and this population genetics actually somehow deals with the study of adaptation, study of evolution. Reproductive genetics, the genetics of reproduction, including genes and chromosome in germ cells and early embryogenesis, developmental biology, and that comes with the developmental genetics. The genetics of normal and abnormal development, including congenital malformations, that we normally call as birth defects. That is the study of developmental genetics, which is a very, very, very interesting area of study, and which is continuously a growing area of study. We need to know several things of our developmental phases, of our developmental stages, to know better as ourselves. Ecogenetics, the interaction of genetics with the environment. Environment controls our gene expression. We will study that. We will study that later in our subsequent classes. But that comes under the broad area of ecogenetics. Forensic genetics, the application of genetic knowledge, including DNA, to legal matters. By this process, we can easily able to identify some killer, some criminal, and that can be a possible way to solve the paternity issues and all that. So now we just need to look around the history of genetics, and as you have already studied some of the experiments of Mendelian genetics and some of the rule of Mendelian genetics in your plus 2 level also, but I want to start the history of genetics from the before Christ era, and on that era it begins with the development of medicine by Hippocrates. who is the father of medicine, believed in the inheritance and acquired characteristics. And to account for this, he devised a hypothesis which is called pangenesis. What is pangenesis? He actually postulated that all organs in the body of a parent gave off invisible seeds. He mentioned the term seeds, which were like miniature component or building blocks and were transmitted during sexual intercourse and resembling themselves in the mother womb to form a baby. This is somehow the beginning of the hypothetical era of genetics. After that, Aristotle emphasized the importance of blood. Still now, we love to emphasize some term like in the blood or in the blood relation, bloodline, and that actually comes from the concept of Aristotle. He thought that blood supplies genetic material or generative building blocks, all parts of the adult body. And he reasoned that blood was the basis for passing on the generative power to the next generation. In fact, he believed that the male semen was purified form of blood and that a woman's menstrual blood was her equivalence of semen. And if these two things combines, that can have the power to produce a baby. But now, after the development of genetics, we know that this concept is actually wrong, but we love to use the term blood relations or bloodlines or in the blood from the idea of Aristotle that still continues. Now comes the era of Charles Darwin. We all know him for his contribution of the theory of evolution. Now, what he actually postulates, his theory actually provides the impetus for scientists to re-examine the question of heredity and inheritance, leading to the rediscovery of Mendel laws and evolution of the field of genetics. His study actually helps us to develop the concept of genetics in a greater extent, and his theory was published from a book that we all know, The Origin of Species, in a short way. The Origin of Species in 1857, and after 10 years of his publication of his book Origin of Species, a scientist named Mendel, and we also love to call him the father of genetics, postulates his study not only in a form of hypothetical way but also based on real experiments, and that is why he referred as a father of genetics. So he was Gregor Johann Mendel. He was actually born to a German speaking family in nowadays the country known as Czech Republic. And in his childhood, he actually worked as a gardener and he studied beekeeping. So this habit of gardening helps him a lot in his scientific pursuit. So when Mendel entered the Faculty of Philosophy, the Department of Natural History and Agriculture in the University of Vienna, no, agriculture, which was headed by John Carl Nassler, who conducted extensive research of heredity, traits of plant and animals. So, upon his recommendation of his physics teacher Friedrich Frenz, Mendel entered into the Augustinian St. Thomas Abbey in Brno and began his training as a priest. And in this period only, he developed himself as a founder of genetic era. So his work was published but actually didn't get any recognition. His work published in the year of 1957, the same year of the Darwin's book. But his work doesn't get any recognition until and unless three scientists independently worked on the same subject, and they are Karl Correns, Erin von Schimmark, and Hugo de Vries. By 1900, research aimed at finding a successful theory of discontinuous inheritance rather than blending inheritance led to independent duplication of Mendel's work by Hugo de Vries and Karl Correns, and the rediscovery of Mendel's writing and laws. So their work actually helped to rediscover the Mendel's work. Whatever they have studied independently, they are not able to get any scientific logic behind their findings. And when they search for a possible explanation of their finding or scientific pursuit, scientific knowledge, they rediscover the Mendel's theory of inheritance or Mendel's study, and mentioned Mendel as the father of genetics. Because if Mendel's study was not happened earlier, then these scientists are not able to define their work. So after that, in the year 1902, the scientists demonstrate that presence of homologous chromosome in cell using dye, which is a field of cytogenetics. In 1905, Bateson provides these studies a title genetics. The genetics was named by Bateson. The same year, geneticists discovered that the homologous chromosome have same size and shape, and the last pair determine the sex, which we all know as a X chromosome or Y chromosome. In 1908, the presentation of Hardy-Weinberg law to explain the gene frequencies of population randomly mating members, we still use the same principle of Hardy-Weinberg law, and this is a very basic principle. And when we study population genetics, this is the main law which we will going to use. In the year 1909, Johnson provides the term gene to unit factors, and after that, in 1910, the Thomas Morgan demonstrated that the inheritance of white eye trait was related to X chromosome inheritance in Drosophila. the beginning of Drosophila genetics, which still now is going on. In 1911, Morgan observed and worked on chromosomal basis for the development of Drosophila. In the year of 1927, X-rays are capable of producing mutations. So from this particular year, we are able to study chromosome, genes, and all that hypothetical things with the help of biophysical instrumentation. And because of this biophysical instrumentation, genetics are able to get its another era. And in this era, several discoveries have been made. So, in the year 1940, Oliver explained the recombination pattern within the gene. We will study the recombination, or you will study the recombination in your molecular biology classes in detail. In 1941, Bedley and Trump presented, and the famous hypothesis one gene, one enzyme concept, which was later explained in a different way in the modern genetical terms. 1944, Avery, MacLeod and McCarthy collectively presented and demonstrated the principle of transformation of genetic material, which is DNA. They are actually able to prove that DNA is our main genetic material. And after that, Martha Chase and Hershey and Chase experiment, we actually call it, demonstrate the genetic material of bacteriophage is actually the DNA. So bacteriophage is a virus. So they are able to prove that in bacteriophage DNA is the genetic material. The main discovery or breakthrough was occurred in the year of 1953. We all know that by the help of X-ray diffraction pattern of DNA, the scientists Watson and Crick are able to decipher the structure of DNA. And in the same year, Binzer published a paper, The Fine Structure of Gene. So this is a very important year for the development of genetics. In the year 1956, Levin presented that the normal human genome consists of 46 chromosomes. Now we know or confirmly know that we have 46 chromosomes. In the year 1957, Franklin and Sange observed and analyzed the storage of genetic information in tobacco mosaic viruses RNA, and after that it was cleared that there are two kinds of viruses available in nature. One is DNA virus and another is RNA virus. After that, Mason-Stahl experiment, which proved the DNA replication models. And in the same way, in the year of 1977, Gilbert and Sanger presented the method of gene sequencing. And still now, different processes of gene sequencing is going on. And now the next gen sequencing process is developed. In 1981, researchers have successfully developed the first transgenic organism. And 1987, Kary-Mullis successfully developed polymerase chain reaction using thermostable bacterial strain, and this is another milestone of genetics. In the year 1995, the era of sequencing or genome sequencing begins by the help of the process showed by Kary-Mullis. In the year 1996, a particular cloned ship was developed by the Wilmot. In the year 1998, researchers are able to develop the full genome sequence of C. elegans. And the full genome sequence of Drosophila melanogaster was completed in the year 2000. And after that, the partial genome sequence or first draft of the human genome sequence was published in the year 2001, and after that the complete human genome sequence was published in 2003, and after that several discoveries are made in the field of genetics. So in this subject or in this course, we are going to study elaborately different parts and different fields of genetics. And for this, you may consult all these books or some of these books. Thank you.